phenotypes is a little bit elusive, apparently, but let's walk through it. So I told you that when this individual, where these genes are very tightly connected on the same chromosome, goes through meiosis, we end up with these 4 gametes. And so one of the classic ways we can set up Punnett squares is we just draw the genotypes of the 2 parents next to each side. And then like you did for the big Punnett square you just made, you drew the 4 possible gametes they make down each side, right? So if I do that, watch what I'm going to get. And actually, I'm going to convert to just using a black pen. Will you guys still be able to tell R's from Y's?
Yes.
So big R, big Y, big R, big Y, little r, little y, and little r, little y. Those are the 4 gametes we just said are produced out of meiosis. You guys agree? And the other parent, what do they make? Well, they're the same genotype, so they make the same set. They're going to make big R, big Y, big R, big Y, little r, little y, little r, little y. Now, you might notice that I made this a little more complicated than I need to, because the reality is these are the exact same genotype. These are the exact same genotype. So if you want to not ever get confused, you can always set it up like I set it up here and just figure out all the possible products of meiosis and then write them along the side here. Or you can recognize that these are exactly the same and these are exactly the same. So the reality is I could just do a 2 by 2 Punnett square. Either way you want to do it is fine. Okay? But if I do it this way, notice what I get. Big R, big Y, big Y. Big R, little r, big Y, little y. Big R, little r, big Y, little y. Little r, little r, little y, little y. So what phenotype are these 3 squares? Round and yellow, right? So these are all round and yellow. What about this square? These are wrinkled and green. And what frequencies then would you recover? Like what would the offspring look like? Round yellow, wrinkled green, and in what frequencies? 3 to 1. So if you have 2 genes that are 2 traits, they're controlled by 2 different genes, and you do a cross like this, and all you recover is the dominant phenotype for both or the recessive phenotype for both, what you have encountered is likely what's known as a linked gene. And so genes that exhibit linkage are genes that, for our purposes, we will just think of as being physically tethered together very close on the same chromosome. So when 2 genes are together very close on the same chromosome, they get co-inherited. So the big R's, because they came from a parent that had big Y's, they get them passed on, they pass them on together. The little r's came from a parent with little y's, so they also get passed on together. So what don't we get that we got in the previous cross here? What's different about the products of this cross than what we did last time? Different ratio, and also, what are we missing? We're missing wrinkled and yellow and round and green. So if you do a cross for these 2 traits and you get a 9 to 3 to 3 to 1 ratio of all of those possible phenotypes, you know those genes are unlinked. If you do the cross and you get a 3 to 1 ratio of just the dominant or recessive type, those genes are linked. Okay? So this is a genetic way to detect essentially physical linkage between genes on a chromosome. Yes?
So is that what you wanted us to in our answer? That they're linked? Or is it because of the independent assortment?
I wanted you to recognize that it wasn't following the rules of independent assortment. And so I gave you, I think I asked you to look up a few things before then it's intended to be a challenging question, all right? And it's not a question that I think everyone should just be able to look at and know the answer to. It's really designed to challenge you guys. And so as long as you take a stab at it, like a real stab, you'll get credit, essentially, for doing it, right? I'm not gonna like penalize you for really trying on a concept we haven't discussed that's pretty complicated and not getting it. I just want you to try to figure it out for yourself, or I wanted you to try to figure it out for yourself. Yeah?
What would the genotype be for red hair and red eye?
Well, the genotype for red hair, red eye, yeah,
For the rabbits.
for the rabbits.
Like, can we make up our own?
Oh, yeah, you can use your own letters. So you could use like H's for the hair and E's for the eyes, however you want to do it. Yes.
Wait, is this what started the whole conversation?
This is what started the whole conversation. Yes.
Everything came together. Okay.
I love when that happens. So then the question is, and this is probably where after we answer this, we need to get started on the lab early today because there's a few kind of techniques we need to show you guys to get this lab to operate well. But the question then that it asks you on the homework is, what would have to happen if you were to recover red-haired, green-eyed, or green-haired, red-eyed rabbits? And the answer would be
That there would have to be the independent
sort of. So let me try to visually represent this for you guys. So I drew a case here that's I intentionally placed the R's and the Y's very, very close together. All right. But imagine a case where they weren't really tightly linked physically, and instead the Y's were way down at the bottom of the chromosome. So there was a big distance of DNA separating them. Now, if I drew a random recombination event onto these chromosomes, if I close my eyes and I kept my eyes open, yes, sorry. I would draw a recombination that happened between the genes. So what would end up occurring if you did recombine is the big Y would move over onto the little r's chromosome, and the little y would move over onto the big R's chromosome. And so then if you, I'm not going to draw it out here, but if you track that all the way through meiosis, you could pop out then with gametes that are big R's with little y's and little r's with big y's. And so the crazy thing about this type of genetic cross is that the closer the genes are, the less likely it is you get a recombination that separates them. And so the more likely you just recover a 3-to-1 ratio of dominant to recessive. When you get a certain distance apart, the odds that a recombination happens between them is so high that you'll recover essentially a 9-to-3 to 3-to-1 ratio of offspring of all the possible types. And if it's somewhere in between, you'll encounter mostly the dominant types for both traits or the recessive types for both traits, and a few individuals that have mixed traits. And those individuals represent the products of a single recombination event. Yeah?
So are they still considered linked since they're on the same chromosome?
I think if you're considering it genetic linkage, if they give you a 9-to-3 to 3-to-1 ratio, I think you would say that they're not linked. I mean, because linkage, there's a number of ways linkage can sort of occur. And so here we're talking about like physical tethering on the chromosome. It's pretty neat. You can really, I mean, if you go on and take genetics classes, you know, you'll get these sets of data where you'll have to use the frequency of individuals that have that mixture of phenotypes to calculate how many recombination events occurred. And based on the number of recombination events that occurred, you can calculate the physical distance between the genes on a chromosome. And back in the old days, before we could sequence DNA, they came up with this measurement called the centimorgan, which is a measurement of how often recombination occurs between them. And the cool thing is that that measure, which is derived just from counting types of offspring, correlates pretty well with like physical distance on the chromosome. So it's kind of a cool experiment.